Right.
Good afternoon, everyone. Thanks for joining us for another of the scientific webinars. Today, we're joined by Armin. Armin is Senior Principal Scientist in the PBPK team, and he has been leading the biologics PBPK science team since 2021. So he's going to be giving a talk today on tissue targeting specificity of antibody therapeutics. It's going to cover physiology and organ structure for biologics PBPK, antibodies, fragments and protein engineering, building, parameterizing and running biologics PBPK models, and a case study in targeting with bispecific antibodies. So yeah, as usual, we will go through the presentation. If people can stay on mute, and we'll record this session, and then at the end, we'll have time for questions. So without further ado, I'll just pass over to Armin, and thank you again for agreeing to present to us today.
Well, thank you very much, Oliver. That's a pleasure and a privilege. Right. The tissue targeting specificity of antibody therapeutics as seen through the lens of biologics, PBPK, and quantitative protein mass spectrometry. Well, I didn't intend this as a deep dive, but it's more like a shallow glide on beta over fairly deep waters. So I'll just remind us briefly about physiology from the perspective of biologics PBPK, that's organ structure, biological processes that matter, organ-specific features, and then a word or two about how we build PBPK models for biologics, how we parameterize them, and it might be useful just to remind us briefly about antibodies, most basically. Well, that's most of the biologic drugs out there anyway. What can be done with them? And then just a few examples about how this biologics PBPK framework can be used, has been used. All right, here we go then. So, yeah, to start with, this human being is neatly split into at least 4 different tissue compartments that we consider. So there are soft tissues like muscle, liver, kidney, and so on and so forth. We also consider nervous system, well, mostly CNS in this context. We also, of course, look at vasculature, which matters a lot, and lymphatics, which is specific for biologics. Right, here we go. Default organ structure, extravasation, FcRn, and organ-specific features. So, just cutting out this small slice of tissue, well, we'll find there are arteries, blood flowing in, blood flowing out. There is oxygen exchange. There are capillaries in between. And in these capillaries, a fraction of the plasma will actually leave circulation. And then it will enter interstitial space, extravascular space. And from that interstitial space, it will then be collected into lymphatic vessels. These flow to the lymph nodes, from lymph nodes through thoracic tract back into venous circulation. So, yeah, vascular capillaries are fairly narrow, as you might imagine. Lymphatic ones are a little bit larger. And believe it or not, this extravasation bit is still hotly contested. 500 years since Da Vinci, we still don't have the final answer. About 100 years ago, a physician, Starling, came up with a hypothesis that, well, at the arterial side, the hydrostatic pressure from the heart drives the liquid into fluid into interstitial space, and most of it is reabsorbed at the venous side, and about 10% then becomes lymph. And that's what every single medical textbook teaches every single student and future doctor to this day. About 20 to 30 years ago, some curious scientists, physiologists started to looking at it more carefully, and it wasn't actually backed up by experimental data. It looked like interstitial pressure was much lower than envisaged in Starling's work at the time. So there is continuous hydrostatic pressure across the entire capillary. Although probably there is a bit more flow from the arterial side, but there is also some flow in the venous side. So in this context, there is only about 4 liters which enters interstitium. But here we go. And that's the format that pretty much each and every single biologic PBPK is following, although I have seen one where this scheme was also applied, but that was the only exception to the rule. So capillaries, they come in all shapes and sizes. We've all heard about the blood-brain barrier, and there is obviously the capillaries in CNS feeding the brain and so on. The endothelial cells are so tightly packed at the junctions, and also then wrapped into astrocytes and so on, that literally no proteins can escape. There is some water flow, sure, but there is virtually no plasma protein escape. Then continuous types endothelial cells in muscle, heart, lung. Well, there are enough paracellular pores that allow some of the liquid fluid to leak out from plasma, and there are even more in kidney, gut, and in liver and bone. Well, the endothelium is very leaky, so that even occasionally cells can just escape just like that. So, as always, modeling is a degree of abstraction. So, and that's how it then is envisaged to happen in the case of biologics and extravasation. We have plasma compartment, we have the tissue compartment in between, they're separated by the endothelial barrier, and there is hydrostatic pressure gradient. So we have proteins of different sizes. Hydrostatic pressure drives them across the barrier. The small proteins will cross the barrier far more easily than the large ones. This creates oncotic pressure difference, which drives which is called isogravimetric flow. It's like a local circular flow. Just adds a little bit to the overall flow, to the flux of the protein rather. And from the concentration gradient, obviously there will be diffusion. So that will be bidirectional, just concentration gradient driven. So that's the basic framework. And the maths behind it was worked out now, what is it, some 6 years ago, bad luck. And that's at the very heart of every single biologics PBPK model that looks at proteins of different sizes. So, and it's as simple as that. Just lymph flow rate, filtration coefficient, Leclerc constant, which characterizes the ratio of filtration over diffusion, and that's that for large pores and small pores. That's all there is to it. But again, there is different equation for each and every organ. There is different equation each and every single protein that we're interested. So the numbers add up fairly quick. And if you try to write this out as a single line of computer code, it does extend beyond the barrier, the end of the paper. So the physiological framework for tube of filtration extravasation was established by Rippe and Haraldson, but they built on the work of many different earlier scientists, started from 50s and 60s. And by and large, organs behave as if there are paracellular pores of two sizes, large ones with a radius of 6 nanometers, and small ones about 4.5 nanometers. So these are the channels for extravasation, filtration, and also diffusion. So it all happens through the same pores. There are no sets of pores which are reserved for this or that. Everything happens all the time through the same pores. And yeah, whenever I've tried to look at it, the experimental data also suggests that if there is a capillary, there is also an antibody seeping out through it, again, supporting the kind of revised Starling hypothesis there. So what does the model then, parameterized model, tell us? The model was actually parameterized with skeletal muscle isolated from rat hind legs, large enough to work. And essentially, they just pumped through that isolated skeletal muscle solutions of proteins of different sizes at different flow rates. So that's how they established the hydrodynamics and quantified the model. So it turns out that the small pores most likely outnumber the large ones by a factor of 8,000 to 1. And yeah, now we can have a look at how proteins of different sizes go through large and small pores. So if it's filtration, then proteins, let's say 100,000, that's roughly, antibody would be 150,000, which pass through without any problems. But small pores will only really let through proteins which are 40 to 50,000 or less. So, and even then it's fairly steep. When it comes to diffusion, again, it's a bit more restricted than filtration actually, but it's the same trend. The take-home message is that large proteins like antibodies, albumin, are extravasated by convection, just filtration. The diffusion contribution is something like 5%. If you think of a small protein, like let's say insulin, for example, that would sit somewhere in this region, that would be extravasated primarily through diffusion. That's why people can inject themselves with insulin, and well, within 20-30 minutes, it's all in circulation. In the case of proteins, it goes through the lymphatics. It takes 3 days before it gets to the circulation. It all makes sense. Right. Yeah, that's a bit of fancy animation there. Right. Up to 2015 or so, you can find papers published along those lines, except that the filtration diffusion parameters were, for practical purposes, impossible to figure out from experimental data, and there was no way to predict them from first principle either. So, yeah, literally within a couple of weeks from each other, we had papers out from Certara and also from GSK, where I worked at the time, where we demonstrated how filtration diffusion coefficients are cross-correlated to the organ-specific lymph flow rate. And all the parameters that go into that correlation are available from the nearest textbook down to the universal gas constant. Well, that would be temperature in absolute kelvins. So lucky enough, it's 37 plus 273 all the time. Then pore radiuses, pressure gradients, well, it's all there. And filtration coefficients can be calculated from protein and then pore sizes. So it's as simple as that. So how do we build the models then? So it's obviously the same very familiar PBPK framework, except that you'll find the green arrows denote here the lymph flow rate. So that is a fraction of plasma going into capillary compartment of every tissue. The fraction seeps out, goes into lymph, and is collected to the lymph node, where it then joins the circulation. So it's by and large the same. Some of biologics models are geared towards antibodies only. What we're talking here is actually fully flexible framework that can accommodate proteins of any size. And yeah, just to be absolutely sure, they can be dosed proteins, let's say from insulin to antibody and beyond. It also covers all endogenous proteins of any size as well. And yeah, just to have a bit of extra spice here, the models also pretty much these days cover mouse, rat, monkey, and human. And in Certara, there are many special populations data for hepatic impairment, renal impairment, and so on and so forth. And the targets can be soluble, can be membrane-bound, so it's flexible. And that's part of the challenge, which means that depending where the target is or targets in the presence or absence of endogenous proteins that need to be considered, the repertoire of models that can and need to be built is very high. There are certain, let's say, standard features which are often carried on and met, but often enough the models need to be customized. So in a PBPK model, we have typical organ structure, which has the vascular structure, has the interstitial structure, has the intercellular space. Unlike in the case of small molecules, intercellular space is out of bounds for large molecules like antibodies. But we do have endosomal compartment, which is in this case assigned mostly to the endothelial cells, which can take up some fluid from surrounding medium, and then where the action with FcRn receptor happens. So as far as this non-specific uptake goes, this is called pinocytosis. It's pinocytosis if it just takes small sips of surrounding liquid and then starts working on them inside the cells. That's what all cells do. It's basic cell biology. Every single cell does it, but some are more thirsty, like let's say antigen-presenting cells. Those take up volume equal to their own cell volume, let's say within a couple of hours. Others are a bit more frugal. And then there is receptor-mediated endocytosis. Cell surface receptors, drug can bind to the receptor. Again, the complex is this causes membrane encapsulation. Again, vesicles forms, and the rest again happens inside the cells. So that's receptor-mediated endocytosis, and that's where TMDD, target-mediated drug disposition, takes place. So what we know is also that, well, in this case we have here, for example, antibody internalized through endocytosis into the endosomal vesicles. That's where it finds or meets the FcRn. Most of the FcRn resides in those vesicles. Small fraction is on cell surface as well. And here the antibody just binds the receptor. What is bound will then be recycled back to either side of the cell, to the luminal or abluminal side. And this also opens route for transcytosis. A small fraction of cells is also antibodies transferred from one side of endothelial cell layer to another, but it's negligible. Sometimes it takes a lot of work to find it out the hard way. So FcRn is not the only receptor that works in this way. There is transferrin receptor, sialyl-glycoprotein receptor, mannose receptor, but those receptors are mostly on cell surface and only a small fraction inside the cells. FcRn, it's the other way around. We don't know exact what's the fraction of the cells on the cell surface of the FcRn, or actually we can't measure the rate constants either. All those need to be fitted from experimental data. And FcRn also binds albumin. So albumin and IgG, that's why those proteins have a quite long half-life. And there is no competition between the two for us as long as we are aware of. There are separate binding sites. A few words about organ-specific adjustments. So in kidneys, we obviously have renal filtration, something like, let's say 20, I think it was around 20% of 10-15% of cardiac output goes to the kidneys. About maybe a third of that goes through the glomerular barrier. So that's a very vigorous flow there. And that filtration step depends on the size of the proteins. Proteins which are smaller than, let's say, 50-60 kilodaltons are filtered out very, very easily and go straight into urine. Water is reabsorbed 99.5%, so what's left behind is then 200-fold concentrated protein, which is the bladder. In the case of brain, we have the same typical organ structure, but in addition to just paracellular, very limited liquid flow, we also have aquaporins. So there is water channels, water pores in fairly vigorous. I think the turnover was in the interstitial space in brain in maybe 3, 4, 6 hours. It's very rich. So protein gets in very, very slowly, but it's flushed out very easily. And as a result, there is something like 300-fold difference between plasma protein concentrations in CNS interstitial spaces and in circulating plasma. It all makes sense there. In case of lungs, we have alveolar space. This can be modeled as simply as passive exchange with very fast local and non-specific degradation, again backed up by experimental data. And in the case of solid tumors, for what we know, there is little to no lymphatics in solid tumors, which means there is no lymph flow. Any exchange is by passive diffusion only. And yeah, vasculature tended to be a bit more porous there. Yep, Felix and came up with very nice model also for modeling subcutaneous intramuscular dosing sites from the mechanistic perspective. So instead of just assigning bioavailability and absorption rate constant, we can just treat the dosing site as a small subsection of skin or skeletal muscle with a bit of draining lymph node that catches the drug. The point here is that the drug goes to the interstitial space and then with lymph is carried to the lymph node where it meets the antigen presenting cells. And antibody concentration in dosed IV dose sample is something 10 times as high than in circulating IgG. So this small dose still has very high concentration and swamps the F7 which is there. And with F7 swamped, it can't rescue from non-specific degradation all of the dose. And some of it goes missing. And yes, as you might guess, that's what bioavailability means in the case of subcutaneous intramuscular dosing. So roughly about 30% goes missing. And that's why it goes missing. And that's where it goes missing. Yeah, and solid tumors. I'm showing this picture because we probably have all seen the onion kind of model where people suggested that there is a circular structure and the outer layer of the solid tumor is all very well exposed, and in the middle of it there is a rotten core of just dead cells and body and fluid. That's not necessarily the reality. It's very easy and comfortable to model mathematically, which is it's a huge bonus. However, when people actually pull out solid tumors, grafted tumors, they find out that they tend to be vascularized through and through, just like normal tissues. Maybe a fraction, while normal tissue would have, let's say, maybe 40 to 50 micron average distance between blood vessels. In tumor, it might be 7 to 8, let's say 60 to 70 or something like that. It's nothing drastic there. So, but blood flow is more irregular and there is no lymphatic. So that justifies treating a solid tumor just as regular tissue, but without lymphatics. Obviously, it's an abstraction, but I think there is a reason for it. So all we have seen now is that there are quite a few features that need to be taken into account. So this happened to be one of the published models where 1 to, let's say, half a dozen different protein species were modeled all at the same time. Some of them also bound FcRn 3 or 4. And you can imagine writing up such a model by hand is certainly possible, but would take a couple of years. And I wouldn't even trust it beyond that, because it would have around 1,300 ODEs and 2,000 something algebraic equation. So it was possible though to write a script that took this template and applied this to the generic layout of organs in physiology, which is conserved across species, and then just assigned the physiological parameters as required. And what you end up then is a network of reactions and compartments, which looks like a cat's cradle on computer screen. And if you zoom in, you can see individual tissues and organs, compartments, all the species, all the reactions, they're all there. You can click on it and everything is where it should be. So yeah, and this is here an example how the two-pore externalization equation looks like for just one species in one compartment. And it is a computer-generated computer code for two-pore exchange. So computer-generated computer model. And thanks to Richard, Tarek, and Abdallah, we also have now the capability to do exactly the same using QSP Designer. So that is a template for generic organs layout. All the organs are there, different dosing sites, so on and so forth. All that needs to be done is to outlining your reaction scheme in this generic visual compartment, and then miracle happens, and within a minute or less, a fully functioning biologics PBPK model is assembled with no extra coding required, on the fly, and meeting exactly the requirements of the project. So it's very easy to, well, obviously, kind of to write up the equations, but we need to put the parameters in. So, and yeah, people every now and then complain that there are too many parameters. It's all too confusing. It doesn't. It's yeah. You can make it essentially. You can prove or disprove anything as you like. So I would argue here that in a typical model there are maybe about 70 physiological parameters. Well, a mouse is a mouse is a mouse. So yeah, one mouse can be different from another, but a pop prep mouse is fairly well characterized as it comes to body weight, organ volumes, blood flow rates, SRM concentrations, so on and so forth. And they are tabulated. They are available from public domain. These parameters are not modified when you code move from one model to another. They are embedded. They are hardwired. Then the second group of parameters which are there are a bit less solid, if you like. These are latent parameters. Most typically, in the case of biologics, could be, for example, organ-specific lymph rates. We can't measure them directly. These are estimated from experimental data. But once they are estimated, they are then locked down. They are no longer modified when we move from one model to another. So again, we have another perhaps 110 parameters, but they are latent parameters. End user typically do not need to tinker with those. And then we have project and target related parameters. So we have antibody or antibody fragment, or you name it. We need to provide affinity, some idea about where the target is, and that's that. And many of those parameters can be measured in vitro, like affinity on by SPR. But there are some parameters which can't be measured easily in vitro, for example, internalization rate constants. But it's not unusual to be able to build a huge model where you actually at the end of the day need to fit maybe 1 or 2 or 3 parameters to run it perfectly well. So yes, the number of model parameters is high. However, you only need to change 2 or 3, actually fewer than in the case of a 2-compartment model. And that's an idea, just an example of how the parameter model can be parameterized for lymph flow rates. So in this case, small protein fragments, all of them inert, were dosed into mice and rats. So there was also albumin inert IgG, and after serial cross sections over several days were taken and quantified by quantitative whole body autoradiography. And something like 50 tissues can be measured, from eyeballs to skin, to lungs, anything. It was pretty amazing. And from that data alone, it's possible then to fit the lymph flow rates, for example. But it's not the only way, but that's one of the options there. The question is, does such rodent parameterized model actually, is it useful, meaningful at all when we need to work, for example, with other species, in this case, monkeys or humans? And yes, it was a relief to see that rodent data parameterized model, in this case, the monkeys were dosed with zirconium-labeled antibodies, which one was with wild-type antibody, the other one with enhanced affinity for FcRn. Again, it was possible to measure the tissue concentrations in living, breathing animals, and it really matters here, I think. And the model predictions were pretty much spot on to the observed data. So the data points are experimental data, the lines are model predictions. There was no tweaking of any kind. So it's possible to extrapolate, hence, from one species to another without invoking any allometric scaling or anything. It also tells you that skeletal muscle in mouse or rat is the same as it is in monkey, or hopefully and most likely also blood-brain barrier is the same, and so on, within margin of experimental error, obviously. And the same is true about humans. Here, zirconium-labeled albumin-binding antibody fragment was dosed into humans. This was purely experimental medicine study. There was no drug coming out of it. They did it just to figure out if the model works. So you can think of it as in vivo labeling of antibodies. So at first, the antibody, the albumin, is all in circulation. Few hours later, it starts to extravasate, and 3 days later, it's pretty much everywhere. In the case of heavy metal ions, there is a small fraction which either leaks out from the chelator or is unchelated in the first instance. So that's trapped almost instantly in the kidneys. It works out around 1 or 2% of the entire dose. It's so black because it's in very small volume. But again, in the grand scheme of things, that means nothing. And again, rodent model predicts perfectly well what's happening in humans. So it is, in the case of biologics PBPK, hence we can have framework which is cross-species and cross-modality, any protein, any size. So what kind of protein-specific parameters we need? Yeah, we alluded already to molecular weight, that's the size of the protein, affinity for FcRn, affinity for the targets. Most biologic drugs are antibodies, most are IgG1, but some are IgG4, I think, as well. But there is quite a bit that one can do with an antibody. It's fairly malleable as a platform. Towards the smaller end, well, we can have peptides, that's semaglutide here, and I would argue that even oligonucleotides like antisense DNA or siRNA fall into this category because they behave like proteins in the sense that they are polar charged, they don't diffuse across plasma membrane, and so on and so forth. And Abdallah has built an excellent, nice paper work out of which has been now submitted and which has collected quite a bit of attention from outside world as well. Now, few words about the targets. Can be soluble targets, mostly cytokines, and about that's 8, I think, are already approved. But you can see, cytokine targets are fairly poorly defined in a sense that this system is all hugely redundant. So it's impossible to hit just IL-5 in one pathway. It will also hit it everywhere else. And even worse, there are cytokines which can just substitute one for the other. So it is fairly difficult to have defined outcome. But yeah, that's the reality. Another class of targets are membrane proteins, typically cytokine receptors or signaling molecules, but there are also others. If it's a BCMA, all it's required is that it is tumor specific. That's that. But we still need to know from the modeling perspective what's the tissue concentration of those targets, their turnover, internalization rate, constant. So how do we get there? So soluble plasma concentrations, plasma proteins can be measured fairly easily. And then, yeah, affinity constants can be measured on biocore or whatnot. But occasionally the number might not be there. So what do we do about it? Fortunately enough, there is a protein mass spectrometric database called PAX-DB. It's public, it's freely available to everybody. And we were able to figure out what's the correlation between the molar concentration of a biologic and its parts per million relative concentration in that database. And that's a value which spans over 5 or 6 orders of magnitude in both directions, from lowish picomolars to high nanomolars. So one can literally just look up the ppm concentration value from the database and make a reasonable guess what the concentration of that protein would be in plasma, instead of having to flow through tens of hundreds of papers. There is some variation here, but I can tell you there is also variation in biochemically measured plasma protein concentrations. Tenfold different values from different papers is very often seen. Depends on the reagent, depends on, I don't know, experimental features which just vary from one user to another. That's as far as tissue plasma concentrations goes. But it's even more challenging or more pressing, actually, more important to have concentration values for tissue proteins, because plasma protein can be just thrown onto a ELISA plate and you have a number half an hour later or an hour later. If the protein is a membrane protein, then that really doesn't work, because the tissue would need to be homogenized, then the protein will still be bound to the membrane. It needs to be solubilized. Then if it's solubilized, it's degraded, it's denatured. If it's denatured, it doesn't bind the antibody anymore. So it's dead end there for all our practical purposes. However, with mass spec, at least it measures the total protein concentration. Just looks at the different peptides and figures out, again, in parts per million units, how much of it is there. And it doesn't discriminate between, let's say, membrane-bound, membrane-embedded fraction and intercellular fraction, if there are such forms, but at least it gives you some idea how much of it is there. Again, and we were able to build a correlation, empirical correlation between known absolute concentrations and measured relative concentrations. So for any protein in this PAX-DB database, we can get reasonably good idea what its tissue concentrations are. And yes, they have data there often enough for most of the tissues and in many different species, from mouse and rat to monkeys and humans. So that covers the target part. Yeah, I alluded to already that MAB is literally, it's a bit like a Swiss Army knife. Yeah, just to remind you, yeah, there are 2 heavy chains, 2 light chains. At the top of the heavy and light chains, there is the binding site called paratope. Every antibody has 2 binding sites for the target. It also binds the FcRn, which is the neonatal receptor in the FC region here. It binds another family of receptors, Fc gamma receptors, in Fc gamma receptors 1 to 3 in the hinge region, if it's an IgG isotype. It also binds complement, and yeah, I can't guarantee it might not bind something else still. So it's a very versatile molecule. And there isn't a nook in the corner of an antibody that hasn't been engineered. And the field is littered with patents, as you can imagine, and people are really suing the daylight out of each other because so much is at stake. So increased affinity for FcRn, no affinity Fc gamma, lower affinity for FcRn, finding affinities at different pH, you name it. Everything has been gone over many times. And antibody is also heterogeneous as a drug. If small molecule drug comes out of the reactor, it's pure. It can be racemic, it can be a mixture of optical isomers. But an antibody would have also oligonucleotides that attach to it, which make it very heterogeneous. And then there will be asparagine glutamines, which will deamidate. There will be more charge heterogeneity. It's a hellish mix. But it is what it is, and that's why manufacturing is as important for antibody as we discover in the end when en route to the clinic. So when we tinker with antibody affinity, we can make it stay in circulation for longer. That's half-life extension. When we tinker with antigen binding affinity, we can make the antibody to capture the antigen from circulation. It's internalized. In endosomes, pH is lower. We can engineer the antibody so that it drops off the cargo there and goes back. That's called pH switching. And likewise, if we make an antibody to stay bound to the FcRn at all pH values, it will be captured by the cells, and it will just carry on circulating, recycling from endosomal compartment. It goes up to the cell surface, it stays bound on the cell surface, captures antigen, and goes back inside again. So that's called pH sweeping or recycling antibody. Right, bispecific antibodies. My God, there is no limit to people's imagination. But don't be fooled. There are only a couple of formats here which work, and as a rule of thumb, the closer it is to normal wild-type antibody structure, the better it works. The only alternative format that makes any kind of use is called diapody. That's behind the bispecific T-cell engagers. Everything else is just fanciful. Right, membrane proteins as cell surface targets. We need to keep in mind that the lipid bilayer is fluid space where proteins can move around. It takes about 1 or 2 minutes for typical membrane proteins to travel enough distance to circumnavigate the cell if they traveled in straight line. This means that if we have an antibody which has 2 binding sites, then within seconds, binding through one site, it will also come into contact with another target on the cell surface, and it will form a ternary complex. Or if we have a bispecific antibody, it will first bind with one arm to one target, or with other arm to the other target, and whilst on cell surface, it will then form the ternary complex, which dissociates very slowly, and that's behind what's called the avidity effect. The limit of it is about 10,000-fold. Experimental data is about less than maybe 10 to 100-fold, but it's very difficult to measure picomolar affinities experimentally on cell surface. And just to mention it, the same formalism would also apply to reactions in cell-cell interfaces. It's 2-dimensional space there. Right, antibody-drug conjugates. Very briefly, the number of attachment sites for the payload depends on the chemistry. If it's N-linked, then it's random to, let's say, lysines and so on. So can be 20 to 30 different sites on cell surface available. Then it's a hideous combinatorial mix. If it's S-linked, then it goes via disulfide bonds in the FC, in the Fab or FC region. Can be taken 2 to 8 payload molecules. However, yeah, at the cost of disturbing the antibody structure to an extent. What people do try to do with ADCs is to make sure that the conjugate has unchanged affinity for the target and unchanged affinity for the Fzer. And the reason is very simple: is if either of these is compromised, the drug will not reach the target, and the payload, which is highly toxic, will just contribute to overall toxicity. So yeah, that was the message here for the ADCs. Now, kinetics, nothing happens instantaneously. Antigen reactions, like any chemical reaction, is ultimately diffusion limited. So that's why typical antibody on-off association rate is around 10 to the 6, 1 over molarity times second. That's where most of them are. So this means that if we have a KD value, we can calculate the off value just by assuming that the K on is somewhere in the region 10 to the 6. There are very, very few antibodies which fall outside this bracket, and if they're substantially below, then I wouldn't touch those antibodies with barge pole. So TMDD, we had work stream about small molecular TMDD that John led, and which also covered the large molecule TMDD. Just to remind you, antibody binds the target, forms the complex is internalized, and the antibody is typically degraded. So this provides additional degradation pathway for that antibody. And then it comes down to the relative contributions of those non-specific targeted, non-specific degradation through endocytosis and specific target-mediated degradation. If the non-specific flux is huge because it is unsaturable, we have linear PK. As the drug concentration drops, the flux through the non-specific pathway drops, and then the relative concentration of the specific pathway increases, and we have faster clearance and shorter half-life. And that's what we see very often in the case of biologics PBPK. In the case of membrane targets, occasionally also with soluble targets. So just a short notice how to compare PBPK with two-compartment model. It still all makes sense. In two-compartment model, the central compartment is pretty much equal to the plasma value, whereas the peripheral compartment in the two-compartment system is based on the assumption that it has the same concentration of the drug as the central compartment. In reality, the extravascular space is about 4 times as large. However, it's about 2 times, about half of it is inaccessible biologics, and in the other half, the concentration of antibody is about half of it would be in circulation. So that's an example how the same data can be fitted to different models, but with biologics, I would argue we get better representation of the reality. And likewise, here we have more or less equilibrium postulated between the two different compartments. In case of biologics, I hope you saw that what we have there is circulation of antibody from central compartment to tissues back via lymphatics. There is no equilibrium there. There is only steady state. Equilibrium in the case of antibody circulation only arrives after death. So, few examples. There is a nasty disease called PNH where people's red blood cells get lysed during the night, so they pass through urine, and it's all full of hemoglobin. And the reason there is an immune deficiency, a protein which protects one's own cells is missing. Hence, the human complement attacks the cells. Fortunately enough, there is an antibody called eculizumab from Alexion, now part of AstraZeneca, which pretty much deals with the issue. It's also the most expensive drug one can, for a long while, it was the most expensive drug, scoring about $500,000 per patient. antibody which blocks a complement factor called C5, which is present at something 600 nanomolar concentration in plasma and has around 60-hour half-life. So there is loads and loads and loads of it. So that it also takes loads and loads of antibody. So 12.5 mg per kg every 2 weeks. Well, that's loads of antibody. So Alexion wanted to make life easier, more comfortable, wondered whether they could come up with something smarter. It's an antibody which has pH switch engineered into it, which I described, and it's also have half-life extension. And yeah, they figured out that when dosing 45 mg per kg, they can drag out the dosing interval to 2 months, which, yeah, from the patient perspective is quite convenient. Now, from the modeling perspective, the question was, could we have predicted the parameters required from first principles only without spending 3, 4, or 5 years in the lab? Could we have just put it down and described what kind of properties do you need to have to reach this kind of dosing interval? So eculizumab at 13 mg per kg blocks C5 for 14 days. So that's this arrow here, and the blue line here, red line here is the C5 concentration. So as soon as C5 creeps back through the barrier, yeah, we need to give it another dose. That's the message here. Just by increasing the dose from 13 mg per kg to 45 mg per kg, we could have extended the dosing interval from 14 days to 35 days, just brute force. Take a larger syringe, pump more drug in, and yeah, you have done half your job. But that's not quite enough. If we keep the dose the same, 45 mg per kg, but engineer pH switch so that the antibody drops off the C5 in the endosome, yeah, that would add another 15 days to the dosing interval. It's still short of 2 months, but it's getting closer. Now, if we factor in the contribution of half-life extension, combine 45 mg per dose with YT half-life extending model affinity mutation, we would get to about 65 days. And if we combine both half-life and pH switch, yes, then it would last for 70 days. And that's really important because 13 mg per kg still left many people who express just more C5 and experience breakthroughs. 45 mg per kg ravulizumab is much more reliable in that context. I don't think it's any cheaper to dose though. So trovimab, that's the role of FcRn. There was huge expectation that FcRn somehow pumps antibody out from circulation into lung interstitium where it will hit the COVID where it hurts. Except that the model didn't really predict it. Model predicted, yes, there will be more antibody in the end because it will be roughly in steady state, but there is not going to be any preferential transport in one direction. So again, the same data, antibody without half-life extension put into monkeys and then studied. And yeah, it was disappointing to the team to see the hard data that the experimental results for different segments of the respiratory tract or tissues for that matter, heart, spleen, etc., were spot on with the model predictions. So proving that there was no additional benefit. Why they assumed it was there are a handful of papers where people measured something in vitro and imagine that there was an effect. In reality, in vivo, there is no effect to be had. There is benefit that the antibody stays in circulation for longer, hence exposure will be higher, but there is no miracle in it. That's what I love. Magic bullet, medicine that kills the target without killing the patient. The first step was not exactly overwhelming, but it's much better. It was much better than there was before, and it holds both for large and small molecules. The expectation. And that's my favorite bit. There are people who say that ADCs deliver payload directly guided by the target specific. That's FDA a year ago, and that's good. Precise design, it brings about the target cell killing, sparing the normal counterpart, and so on and so forth. It looks like these people live in one kind of universe. And then we have another universe where people point out that off-target toxicities are a major obstacle to ADC therapies. And one of the downfalls of ADCs is that you really struggle to get them into solid tumors and so on. There couldn't be starker contrast between the two. So where are we then? If we model, compare two popular tumor targets like HER2, EGFR, you can figure out the tissue concentrations. Where does the antibody dose go? That's the end question here. And yep, by fitting the data, just plasma concentration to the internalization rate concept for both antibodies, we can figure out what the tissue concentrations are from the model, and the results indicate that the antibody dose distributes between different organs pretty much in line with organ size. Tumor collects around 0.1% of the dose, and that's actually also in agreement with experimental data, and there is not that much difference between EGFR targeting cetuximab and Herceptin, HER2 targeting Herceptin. And what's even more telling is that tumor takes small fraction, but it takes fractionally more per volume. So there is benefit certainly to cetuximab, Herceptin, or respective ADCs, but the difference between healthy tissue and target tissue about 3 to 4-fold. So that's the width of the therapeutic window, and that's why it's so difficult to dose an ADC to the patient without killing it before the disease does. Hopefully, there are many who still will be saved, of course, in the end, if dosed very carefully. And we can do the same exercise this time a bit more in silico, to see if bispecific antibody can help. I'm using actually the same EGFR HER2 data, but I'm modeling here a hypothetical antibody that would be bispecific, can bind with one target or the second target, or in cells which have both targets by one arm or both arms, or cells which don't have any, will still be internalizing a little bit. So as far as plasma PK goes, the bispecific antibody would probably be cleared more quickly because it can engage more target, makes very common sense. And as the dose decreases from 10 mg per kg to 1 mg per kg, it would be gone within a couple of days from circulation. Now, how does it impact tumor engagement? Well, it was fairly surprising to see that the difference here, for example, on tumor cells between bispecific antibodies, this one here, and monospecific antibody is actually very narrow, and it only happens when the plasma concentration of circulating antibody is very low. The reason behind it is that bispecific antibody will also bind through one arm, and specificity for bivalent engagement only arises when the concentration drops to picomolar concentrations, and that's another problem there, because at picomolar concentrations there might not be enough target engagement to deliver the drug. So these are the physiologically imposed boundaries to antibody targeting. So bispecific antibody can be more specific than monospecific, but it's not a miracle drug. It's the same as with monospecific antibody, just a fraction goes to the targeted organ, something between 0.1 and 1%, depending on the dose and the target expression level. And we can have specific targeting, reaching antibodies, reaching the tumor cells in solid tumor. At the lower the dose, the better. But again, the antibody will also be exposed to the target in circulation. If the target is on endothelial cells, those cells will get a real hard hit as well. So, have we found the magic bullet yet? I don't think so. Shall we find it anytime soon? I don't think so. The reason is that target-specific binding of antibodies in vitro doesn't mean tissue-specific delivery of antibody in vivo. And but it's not entirely hopeless in the sense that up to a point, one can still vary the affinity of the antibody to maximize the drug taken up in the system. And that's where the modeling comes in. So 3 pillars. I don't need to convince you that it helps to do the right drug discovery. And just the last slide to leave a bit of a question. We have, for example, in the case of small molecule drugs, we have KB, just the fusion limited model. We also have now permeability limited models for small molecule drugs. As you saw, the large molecule models are all permeability limited. So maybe the next question is, can we also engineer the small molecule features to the large molecule model to see if there is a unified framework? And that was the last slide. Thank you very much for your attention.
Thank you, Armin. I don't know if there's any quick questions. I know that we've reached time. Don't see any. Thanks, Armin. I know you've got a train to catch as well. So
yeah,
I appreciate you taking the time to do the presentation today. And just a reminder that there'll be registration for further workshop, further presentations, which are coming up online shortly for the rest of the year. Okay, thank you, everyone, and take care.
Thank you. Good night.